Hello students, welcome to EPG Pathshala of University Grant Commission India. I am Dr. Sohail, an assistant professor from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard, New Delhi. Today we are going to discuss about a module that is a specialized osmotic drug delivery system from the paper Novel Drug Delivery System 1. My discussion will include this is basically the osmotic. It is a part from the osmotic drug delivery system, and my discussion will include osmotic bursting osmotic pump. We will discuss also about control porosity osmotic pump and liquid oral osmotic system, and we will discuss also about oral osmotic system clone targeting called as OROSCT. And the final objective of the discussion is will include the sandwiches oral therapeutic system. So this is also a kind of osmotic control drug delivery system. So let's start with the first one, osmotic bursting osmotic pump. This delivery device is similar to that of elementary osmotic pump EOP, except the orifice in this case is absent. So as you remember from the past that in every osmotic drug delivery system you have a important component called orifice which is meant to release the drug. However, in this case osmotic burst osmotic pump, there is no orifice. So this is the basic difference. Now the core part of osmotic burst osmotic pump OBOP is comprises of a drug and an osmogen, whereas the coarse part is semi-permeable membrane. When it is placed in an aqueous environment, water is embedded and hydraulic pressure is developed inside the device to a limit until the wall rupture and the incorporated drugs are released to the environment or to the biological fluid. In this case, the release of drug is controlled by varying the thickness of the membrane as well as the surface area of the semi-permeable membrane. This is also from the category of oral osmotically active controlled drug delivery system. So, and this one is controlled. It is based on the controlled porosity osmotic pump. It is made up of a core, which is basically the drug core, a semi-permeable membrane containing water-soluble additives, for example amino acids, sorbitol, dimethyl sulfone, etc. Okay, and that can be capable of producing a pore size of 10 angstrom to 100 micrometer. We had already learned before that we can create pores not mechanical way but just with the help of some chemicals that can dissolve, okay, and create certain pore. This is what we are discussing here. So now the drug core surrounded by a symmetric structure membrane comprising of a thin dense skin layer supported by a porous structure. The membrane is permeable to the water but impermeable to solute. Okay, so when the membrane come in contact with the water, low level of water soluble additives are released from the polymembrane, and that where actually the pore created, and it is permeable to the water but impermeable to the insoluble solutes. So finally this resulted in the formation of a sponge-like structure which was permeable to both water and dissolved drug substance. So water enters the core through the microchannels and form a saturated solution of drug. Again, the saturated solution case is here, so it means drug will move from high concentration zone to low or no concentration zone of the solvent. So now diffusion takes place, the drug is released in the controlled manner. Here the rate of drug delivery from the structure depend on area of coating, okay, the water permeability of semi-permeable membrane, osmotic pressure of the core formulation, and the thickness and the total surface as well. The rate of flow of water into the device is governed by the formula dV upon dt, that is called the rate of flow, equal to AK upon H dP minus dr. So this dV upon dt is the rate of water flow, K is the permeability across the membrane, permeability constant, A is the area of the membrane, dP is the osmotic pressure difference, and dr is the hydraulic pressure difference. Here is the structure diagram showing before the operation, the formulation, during the operation, how it look like. So as you can see, the rigid semi-permeable membrane, and then the core is osmotic drug core. The semi-permeable membrane contain the material that can be solubilized or dissolved in the presence of biological fluid. So when the biological fluid come in contact, you can see it create the pores. It forms the membrane like a microporous membrane. So those portions which contain the mixture of rigid semi-permeable membrane and this kind of material, after the material get dissolved, only the rigid membrane remain, and ultimately you have a porous structure. Let's discuss about liquid oral osmotic system. These are designed to deliver incorporated drug as liquid formulation, including self-emulsifying formulation that could be self microemulsified form, self nanoemulsified form, or the combination the benefit of extended release with high solubility. This liquid oral osmotic delivery system can further categorize into three subcategory or three subtype. Uh, LOROS hard capsule, LOROS soft capsule, and delayed liquid bolus delivery system. All this system include a drug layer in liquid form, then osmotic layer, and finally the outer layer is semi-permeable membrane. When such system come in contact with the aqueous environment, water permeate across the rate limiting semi-permeable membrane, and then it activate the osmotic layer within, which further push the drug or release the drug by the high differences osmotic pressure. The expansion of the osmotic layer resulted in the development of hydraulic pressure inside the osmotic delivery system, thereby forming the delivery of liquid formulation through the orifice. So the LOROS hard capsules or soft capsule system are designed to provide continuous drug delivery through the orifice. So this is another kind of design. This is different than the tablet design or bilayer tablet design, which is also one of the example in osmotic control or drug delivery system. Going into the detail of this design, the L shape or LOROS delay liquid bolus of drug delivery system is designed to deliver a pulse of incorporated liquid drug. So drug is mostly in this case is a solubilized form. The delayed liquid bolus delivery system has a unique design which comprises of three distinct layers, which include a placebo or delay layer, a liquid drug layer, and osmotic engine. So you will learn later about all these layers. All these layers are surrounded by rate controlling semi-permeable membrane, which is a common like any other osmotic control drug delivery system. So now the device orifice is drilled on the placebo layer end of the capsule shaped delivery device. The delivery orifice is on the top of the capsule layer. So when the osmotic engine is expanded, the placebo release first followed by the delayed release of the drug layer. Drug release can be modulated to be delayed from 1 to 10 hours, depending on the permeability of rate controlling semi-permeable membrane and the thickness of the placebo layer. Here you can see two structures such design and their components. You can see one is egg-like structure and other one is more or less like a square cell tablet structure. Basically these designs are used in the form of capsules. Delivery orifice, it consists of the egg-like structure, it consists of delivery orifice, and then rate controlling membrane, which is semi-permeable membrane. Core is liquid drug formulation, so drug is dissolved in the liquid, and outside of this core, just adjacent to the core, it is osmotic push layer, and then there is a barrier inner membrane. In the other structure you can see it's the same kind of a structure. There's a two compartment 1 and compartment 2. Compartment 1 is the drug compartment 2 is another drug compartment, so two drugs which are separated, and the third compartment is push compartment. At the top it is the delivery orifice, it is covered with the drug overcoat, and then a rate limiting membrane is there. In the oral route, the osmotic control drug delivery system delivered by oral route. Another example in this category is OROS-CT. CT means colon targeting. This device is specially designed to release the drug in colon. Colon is the part, as you already know, this is the part of intestine, basically the large intestine, which is comprises of cecum, colon and rectum. So it is the middle part of the large intestine. This is used when majority of drug or any drug which are really gastric irritant to save the and may lead to the gastric ulcer. So this is used to deliver the drug directly to the colon. In some cases also we need to deliver the drug directly to the colon, like in the situation or condition called ulcerative colitis, where you need to target directly the colon with the drug. So it is 5 to 6 enteric coated push-pull osmotic unit are combined together in this system and filled in a hard gelatin capsule to fabricate, that is filled in a hard gelatin capsule. When this device come in contact of gastric fluid, hard gelatin capsule dissolve first, and this stage enteric coating check the fluid entry into the device. So actually dissolution, separation of layer or dissolution of top layer of the capsule is started in the stomach region. However, it also further have the coating. That coating is enteric coating that protect the inner core at this stage, and only started dissolving in the colon. The enteric coating get dissolved in intestine, yes, in alkaline pH, and due to concentration gradient generated across the semipermeable membrane, water into the system here now. This water imbibition by the polymeric osmogen resulting in the swelling of push compartment, and simultaneously gel formation occur inside the drug chamber. The drug formulation is then pushed out of the device by swollen polymer. Okay, so this device is capable of targeting the colon because it consists of 5 to 6 units of push-pull osmotic pump, which release the drug one by one. Obviously, it has a coating layer within the capsule. That coating layer is only meant to dissolve or made up of a material that dissolve in the intestinal pH. So this is a basic diagram of this colon targeting osmotically active or osmotically regulated drug delivery system. You can see it is the layer by layer structure, but and it is same. So just I am showing you the structure of one in magnified form. You can see it has orifice, and then after the orifice, this orifice is created on the top of the semipermeable membrane. In the core there is the osmotic drug compartment. The core is separated into two parts: osmotic drug compartment and then the osmotic push compartment. The outermost layer is also an enteric permeability membrane. This is another example of oral osmotic control drug delivery system. It is called sandwich oral therapeutic delivery, abbreviated at SOTS. So in this device, polymeric push layer is sandwiched between the two drug layer and coated with a semi-permeable membrane. As far as the fundamental structure is concerned, compared to the other, it resembles push-pull osmotic pump. So the basic structure is same, just like basic push-pull osmotic pump. Each drug layer has its own delivery orifice opposite to others. So except than this, in other previous cases, what we have noticed that you have a single orifice either on the top or somewhere in the middle of a separating layer. However, in this sandwich one, each individual's layer have its own orifice. So when imbibition occur, that is water come inside and dissolving the osmogen and creating pressure, inside the gastrointestinal tract, polymer layers swell to push the drug out simultaneously from both the drug layers. This is how it works. So this is the basic structural diagram indicating before operation and after oral intake how it works. So you can see before operation it looked like there is a two orifice, semi-permeable and orifice, and then it is a semi-permeable membrane with plasticizer for other benefits. Two separate drug layer, two separate drug layer is there. Definitely there will be two orifice for both layer, and then in the middle there is a push layer, which is the polymeric layer. So when water goes in, this push layer of polymer starts swelling, When the polymers swell, the dissolved drug in both the layer is coming out from both the orifice. This is how it works. Now, another important type of osmotic system is monolithic one. In this system, a water-soluble drug is dispersed in a polymeric matrix, and then further it is coated with a semi-permeable membrane. Here we are using the abbreviation for semi-permeable membrane, SPM. Okay, so when the system come in contact of a aqueous environment, imbibition of water or absorption of water takes place by the drug uh, within the matrix system. This polymer matrix then further rupture, drug release happen when the hydraulic pressure inside the device exceed a certain limit. Such a monolithic system is different from the osmotic bursting osmotic pump because it does not contain any osmotic agents. Rather, polymeric matrix is used to start the osmotic phenomena in the system. Now, osmotic matrix system, abbreviated as OSMAT. This is relatively a newer osmotic controlled drug delivery device which exploits the property of hydrophilic polymers. So basically it is designed with the help of hydrophilic polymers. These polymers swell and form a gel when placed in an aqueous medium, and also further responsible for generating an in situ semi-permeable membrane. Therefore, the used polymer in this system are meant for the development of a matrix system and also simultaneously operating the functional similar to semi-permeable membrane. This is another example of osmotic pump controlled drug delivery system. It is called Telescopic Capsules, and it is also used for delayed release. Delayed release simply means there is no release or onset of release until a certain time point, and the release is like immediate or in controlled fashion after that time point. How it different? In some formulation, the release started right after intake, that is, you can say the release started immediately at 0 point or right after the intake of certain small lag. However, in delayed release there is a significant and different time lag. Maybe it is of 1 minute, maybe it is of half an hour, or maybe it is in of many hours or in a day. So let's discuss the telescope capsule for delayed release. This device mainly consists of two chambers. First chamber contains the drug as well as the exit port, that is orifice to release the drug. The second chamber contains an osmotic engine, which is generally a layer of wax type material which separate the two chambers. Next important point with this structure is that to assemble the delivery device in a functional manner, the desired active agent is usually placed into one of the chamber by manual or automatic fill mechanism. So osmotic chamber in the system contain the bilayer tablet. It consists of bilayer tablet actually, and it is placed into the complete cap part of the capsules, and the bilayer gets exposed toward the cap opening. Finally, the fluid so what actually happening in the system is that as fluid get imbibed the housing of the dispensing device, the osmotic engine expand and exert a considerable or higher pressure on the slidable connection or connected to the first as well as the second wall connection. So during the delay period, the volume of reservoir that contain the active agents is always remain constant. Hence, a negligible pressure gradient exists between the surrounding environment and the interior of the reservoir. Finally, as a result, the net flow of environmental fluid derived or driven by the pressure entering the reservoir component is minimum, and consecutively no agent is delivered for a certain period, and that is how they achieve the lag time. Now, under the category of newer technology in osmotic control drug delivery system, if you just recall your past slides in osmotic control drug delivery system, you learned about implantable one, you learned about the oral, and I told you there in the classification there is a separate category called a specific or a specialized one. So this is that one, and these are the newer technology which is now used in this system. They are osmotics technology. And what is this? The Osmodex family of technology is combine the laser drill tablet technology along with the variety of single active or multiple active drug delivery devices. So in Osmodex system resulted in simplifying the dosing and the adds in proper patient compliance. The another one under this newer technology is Osmodex ID delivery for insoluble drug. These kind of devices can accommodate first order, zero order, or delayed release mechanism while releasing the incorporated drug over predetermined time frame. This technology has been used to solve the multiple challenging insoluble drug delivery problem. Example includes osmotic nifedipine extended release tablet, which is a common tablet formulation in the market using this technology. Nifedipine is well known. You already know that this is a drug used in cardiovascular disorder, and it is an example of calcium channel blocker. So it is a prolonged acting or control release calcium channel blocker formulation, and it is applicable or used in the disease like arrhythmia, and it also used in case of essential hypertension. Is about osmodex ID delivery was soluble drug. So this is actually again a oral control osmotic active drug delivery system, and it is basically designed for water soluble drugs. So this platform technology can be utilized efficiently to resolve delivery challenges associated with the low soluble or low viable drug or drug requiring a targeted over a time or targeting over a time. It involves a combination of immediate release as well as control release, either one or two drugs. So combination drug delivery can be achieved, yes, with this system. Another one is that it is composed of immediate release component or immediate release system and then a control release, both part. Example of this is Allerga D224. So our tablet, so it is from Allerga, is a company that is manufacturing this particular technology. The fourth example is Osmodex double CR combination. As the name itself indicates, it is a double control release combination. So this is dual control release platform allows sufficient delivery of two drugs simultaneously from a single osmotic tablet, where each drug release pattern of the two different drugs can be independently modulated to the desired release pattern. Again, so these two systems that you learned here or that you came to know here are basically can be designed for delivery of two drugs simultaneously. Now come to the last category. So you already in the beginning you learned that this osmotic active drug delivery system comes from three categories. One is implantable, another one is oral. Now this one is the last. It is a specific delivery devices. Examples are Doros technology. So what Doros technology is? These are the specific miniature osmotic implants which deliver the incorporated drug for 3 months to 1 year with zero order control release kinetic. So it is implant. So it should come, or it can be also an example of implantable or injectable devices. This technology is well suited for potent drug which has the capability to deliver up to approximately 500 milligram of drug from a single implant. Now, optimization of various formulation technology has led to the development of formulation that maximize drug payload, in this case, stabilize the drug chemically within the system, physically from extended period of time at a body temperature, and then the drug formulation reside into the drug reservoir component. So these are already an optimized system available in the market. The drug formulation may be either in the form of solution or suspension in this system. This can be both drug solution and can be both aqueous or non-aqueous in nature. So drug solution, either lipophilic drug or hydrophilic drug, both can be incorporated in this. Okay, Doros drug formulation exhibits appreciable stability at body temperature for extended time period, Usually it ranges from 3 months to 1 year. Doro system offers suitable biocompatibility. It is well suited for implantation purpose. So now the next one is the targeted drug delivery system with the catheterized osmotic pump. So it is used with the catheter. So catheters of various design can be attached to the exit pole of the osmotic pump for targeted drug delivery system. These devices should be flexible, yes, compatible with the targeted tissue or organ, and non-reactive and non-absorptive toward the drug solution itself. So the most common used material for such catheter are silicon elastomers, polyolines polymers such as low density polyethylene. So under the specific drug delivery using Doros with a precision miniature catheter. So this is under the catheter system. So in this one, for targeted site drug delivery, direct is catheter technology which can be attached to the Doros. So these are the patented technology, and that's why you see the name typical to that particular company. For targeted site delivery, direct is the catheter technology which is used in this case, which is attached to the Doros system to deliver directly the flow of the incorporated drug to the targeted organ and tissue. Targeted site specific delivery enable the therapeutic concentration of drug to that particular region without exposing the entire body, okay, and therefore off targeting is minimum. So drug toxicity in other organ is not going to happen, or it may be very minimal. So this is the advantage. So therefore the miniature size, the performance efficiency property of Duru system allows for a continuous site specific delivery to number of precision location within the body. Example of such device include intrathecal opioid delivery system, which is indicated for a number of chronic pain conditions such as chronic back and leg pain, chronic cancer pain, and painful neuropathy. So here we are going to discuss about Doro's intratemporal delivery of anti-neoplastic agents into the brainstem. So this is basically delivering of cancer drug, cancer chemotherapeutics to a local or targeted site. So therefore the concentration in that particular cancer region or tumor region will be high, and it will stop or reduce the drug delivery or drug concentration to other healthy organ. Therefore the associated toxicity with the anti-neoplastic agents or anti-cancer agent is reduced in this case. So it increases the safety for that particular drug, and also reduction in dose can be possible. So increase in appreciable survival rate were demonstrated in the past with such kind of system. In the studies we have figured out already. The brainstem continue monitor and regulate the cardiovascular, respiratory, and other automatic functions, which resulted in targeting the chemotherapy directly into the brain area. So this is developed particularly for right now for the brain tumor. So far we have gone through many advantage, different kind of and preparation techniques with the osmotic drug delivery system. We learned a lot of advantage like patient compliance, control delivery, reduction in dosing frequency, and we can also reduce the side effects by making such kind of delivery system. However, the system also have some disadvantages. So in this slides we are going to enlist you or tell you some of the side effects with disadvantage of this drug delivery system. First of all, the cost of the formulation. In compared to the conventional dosage form design like tablet and capsule, osmotic devices design are relatively costlier because their fabrication need lot of effort and time, and also components as well. Another issue that this kind of dosage form may face is the dumping. The dose dumping or irregular or uncontrolled release of drug from the device is also, that is the part of the dose dumping. Another issue that can be faced by such osmotic drug delivery system is gastrointestinal irritation. Ultimately, when the drug come out from the system, maybe the release is more, and in a single time point major fraction of drug or high concentration of drug comes into the GIT tract, and it exposed, GIT tract is exposed by the by such drug. If the drug is gastroirritant, which is commonly seen in the case of a non-steroidal anti-inflammatory drug category, it may cause irritation and ultimately it may lead to the ulceration in the GIT tract. Some other disadvantage with osmotic drug delivery systems are uh, they are sometimes not very effective, due to gastric motility itself. And last but not the least, that the diameter hole is very important in such design and always need a special equipment to create that delivery orifice. So here are some of the important formulation factors that affecting the drug release from oral osmotic drug delivery devices. You already had learned what are the factors that responsible or affect the drug release from implantable one. This is the factor that affect from the oral one. Drug solubility is one of the important factor. Release rate of encased or loaded drug from the device is directly proportional to the drug solubility within the core. High water soluble drug as well as poor water soluble drug are not a good candidate while making osmotic drug delivery system. So therefore it should be moderate and there must be a range which I am going to discuss later. Hence a number of approaches should be employed to deliver drug having property of extreme of solubility. Suitable candidate for this is any drug that is having water solubility from 550 to 300 milligram per ml. Next, by properly modulating the solubility of the drug within the core, effective release pattern can be achieved. So yes, we can modulate. What do you mean by modulating drug release? Here you can change the drug property by making prodrug, and with the help of this prodrug you can decrease the solubility of highly soluble drug, or you can increase the solubility of poorly soluble. The various approaches to achieve this co-compressing of the drug within the excipient with modulate the solubility of drug within the core. This includes other way of playing with the solubility is the use of soluble polymer in the core which swell after absorbing the water in the surrounding environment and that push the poorly water soluble drug from the orifice. Next technique is effective use of effervescent mixture to increase the release of poorly water soluble drug from the orifice. Effervescent mixture are what? These are the chemicals when it dissolve, it release the CO2 carbon dioxide and started making bubbles, and that help in creating the pressure to release the molecule or drug molecule from the orifice. Now the last one is to use various cyclodextrin derivatives. Cyclodextrin is a well-known technique to improve the solubility of poorly water soluble drug. Other techniques you can say use like nanosizing of drug, reducing the size of the drug also help in improving solubility. Making the nanoemulsified form of the drug is also improving the solubility. You can add some sort of surfactant with the drug mixture that will help in further improving the solubility. So as we had discussed about the basic principle behind the osmotic active or osmotic controlled drug delivery system, we know the basic principle is osmosis. So to understand osmosis and the outcome of osmosis, that is osmotic pressure, is very important to understand and to correlate it with the drug release from the system. The rate of drug release is directly proportional to the osmotic pressure of the core formulation, and the drug release do not possess adequate if the drug do not possess adequate osmotic pressure, we have to add or additional osmotically active agents should be added to the formulation. So because it is ultimately and completely depend on the concentration of osmotically active agents, and the most simplest and most predictable process to achieve a constant osmotic pressure within the formulation or within the device is to maintain a saturated solution of osmotic agent in the compartments. Another important component of osmotic active or osmotic drug delivery system is delivery orifice, that is the pore from where the solubilized or dispersed drug coming out from the delivery system or device under the high osmotic pressure, that is the delivery orifice. This delivery orifice should be within the desired range for control and controlling the drug release from the device. Now, another component, that is the coating membrane. Uh, the release rate of drug from the osmotic device is also affected by the number of characteristics of coating membrane, that include thickness of the membrane itself, type and nature of membrane forming polymer, as well as the presence of other additives like plasticizer is one of the example. The membrane permeability of device can be modulated. Yes, we can increase or decrease by proper choice of membrane forming polymer, and at the same time we can add some additives. As I already told you, plasticizer is a common additive which is used to include within the polymer. So now let's summarize this chapter, this particular module, what we have learned in this. In this particular section, we learned that osmotic active control drug delivery devices can be categorized in different category based on their mode of application. Oral, injectable, also can be categorized on the basis of mechanism by which they are releasing the drug. Under the oral category and under the injectable category, they are of different kind. In this particular segment, we studied more about the specialized kind of osmotic active drug delivery devices. We further learned that it is actually the orifice, drug delivery orifice, and semi-permeable membrane thickness, and the concentration or type of plasticizers, and also the concentration of the osmogen within the device, altogether playing the role in controlling the drug delivery or release of drug from the device. Moreover, the desirable drug release kinetic for oral delivery, which is the zero order release kinetic, can be achieved by playing with these particular components and their concentration while making the while making the osmotic control drug delivery devices. We had further learned some advantage with such control drug delivery devices, but on the other hand, these devices has its own shortcomings. The most common or simplest one I can tell you here is that compared to the conventional dosage form, which is tablet and capsule, designing of these kind of dosage form is relatively costlier. This is one of the important one. And for the oral delivery, if you are using these kind of devices, cannot eliminate the problem if the drug itself had the gastro irritation kind of issues. Dose dumping may happen or may not. So it depends. These shortcomings can be eliminated by playing with the formulation parameters. So this is all about osmotic active or osmotic control drug delivery devices. Thank you so much for your time and listening.